Hi, this is Priya N. Again signing in to the next lecture, Atomic Absorption Spectroscopy. To start with, Atomic Absorption Spectroscopy. It is based on light absorption and emission in gaseous phase. It is similar to flame photometry, but flame photometry measures the intensity of the emitted light when an activated atom returns to the ground state, and it measures the absorption of light of an unique wavelength by atoms in the ground state. And this spectroscopy is used to identify and determine the concentration of specific elements in the sample, and it was first invented by Alan Bosch in 1955. Let's move on to principle. Principle consists of a monochromatic light for a particular element is produced by means of a halo cathode lamp, utilizing that element as a cathode. And this monochromatic light is beamed through a long flame into which the solution is aspirated to be analyzed, and the heat energy dissociates the molecules and converts the components to atoms. Although some atoms are activated and most atoms remain in the ground state at the temperatures commonly used. The transmitted light that is not absorbed reaches the monochromata, which passes only the wavelength close to the resonance lines of the particular element to be assayed. And the transmitted light strikes a detector, and the decrease in transmitted light is measured. And this one is the atomic absorption spectroscopy diagram, and it consists of halo cathode lamp, atomizer, monochromata, nebulizer, sample, detector, and recorder. So here atomizer, what is the role of it? Elements to be analyzed must be in atomic state, and the atomization is separation of particles into individual molecules and breaking molecules into individual atoms. Okay, so this is done by exposing the sample to high temperature in a flame or graphite furnace. Then the light emitted or absorbed by the atom in the vapor state generates signal which is proportional to the concentration of the sample. Then components of atomic absorption spectroscopy. It has halo cathode lamp, laminar burner, monochromator, photomultiplier tube, and readout device. Halo cathode lamp. It is nothing but it is a light source. The lamp is filled with an inert gas like argon or neon at low temperature. And when the lamp is fired, gas atoms between the electrodes are ionized and strike the cathode with high velocity, causing metal atoms to be sputtered from the cathode surface. So what is meant by sputter? The radiation source is a halo cathode lamp. It consists of tungsten anode and a cylindrical cathode, scaled in a glass tube containing an inert gas such as argon at a temperature of 1 to 5 torr. And the cathode is fabricated usually from the metal to be analyzed. And the applications of a potential of about 300 volt across the electrodes causes ionization of the organ and generations of a current of 5 to 10 milliangstrom as the organ cations migrate towards the cathode and electron migrate towards the anode. So if the potential is large, the argon cation strike the cathode with sufficient energy to dislodge some of the metal atoms and produce an atomic cloud. So this process is said to be sputtering. This one is the halo cathode lamp, consists of base plug, cathode, glass shield, anode, glass envelope, and window. This atomic absorption spectroscopy differs from emission flame photometry in having a radiation source. It is practically impossible to isolate a particular resonance wavelength from a continuous source by using a prism or a diffraction grating. So this problem has been solved by the development of halo cathode discharge lamps. Such lamps produce monochromatic radiation characteristic of the element to be analyzed, and in this lamp the cathode is a halo tube which is lined by the element in question. The lamp will thus emit monochromatic radiation characteristic of the emission spectrum of the element involved. Such lamps have now become commercially available for a long range of elements. So it is the second unique component in the system. The burner consists of three parts, as I told you earlier in the previous slide. It is nebulizer, premix chamber, and burner head. A sample in solution is aspirated and nebulized, which is reduced to a fine spray by a stream of oxidant, that is air or oxygen, flowing across a sample capillary tube. The mist is mixed with oxidant and fuel. The fuel will be here as acetylene in the premix chamber, where larger droplets are trapped and drained off. The mixture of gases and sample is directed into the burner head and the flame. The burner is especially one for atomic absorption spectrometer. It has a long, flat, topped head positioned directly below and parallel to the beam of light from the lamp. Then the gas flow through a 10 centimeter long slot at the top of the burner head so that a long thin curtain of flame is produced. Then analytical procedure. The sample to be analyzed or prepared in the form of solutions. Plant and animal tissues are first ashed by wet or dry ashing techniques, and then a solution of ash is prepared in hydrochloric acid. Then this solution is finally diluted with water to appropriate concentration, and the solution of sample in oil or other non-aqueous liquids are prepared in organic solvents such as methyl, isobutyl ketone, etc. And the halo cathode lamp for the required element is put on for 30 minutes for warming up. The correct monochromator wavelength is selected and the slit width is adjusted to isolate the line to be measured. The readout is adjusted to isolate the line to be measured, and the readout is adjusted to zero transmission when the light is cut off from the detector, and when the blank solution is aspirated, the readout is adjusted to percent transmission. The standards and the sample are aspirated and the readings are taken. A plot of concentration against absorbance gives almost a straight line. The amount of metals present in the given sample is calculated from the calibration graph. Monochromators and detectors. The function of the monochromators is to block light wavelengths different from the desired resonance line from reaching the detector. Most instruments have a good quality ultraviolet and visible monochromator. Photomultiplier tubes are used for the conversion of the radiant energy signal to an electrical one. Readout devices. The chart recorder is a potentiometer using a servometer to move the recording pan. The displacement in the pan is proportional to the input voltage. Then advantages. It is more sensitive than emission flame photometry. Minute amount of an element can be determined in the presence of high concentration of other elements. It is highly specific because the atoms of a particular element can only absorb radiation of their own characteristic wavelength. And disadvantages. There is a need of separate lamp source for each element to be determined. The use of this technique is limited to metals only, and certain metals such as aluminum, molybdenum, etc. cannot be detected because they form corresponding oxides in the flame. Thank you.